Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1953 witnessed a profound shift in our understanding of life's fundamental building blocks, a revolution propelled by the meticulous research of Francis Crick and James Watson. Their seminal publication, Molecular Structure of Nucleic Acids, not only elucidated the double helix structure of DNA but also inaugurated a new era in molecular biology. This essay will delve into the genesis and enduring legacy of this transformative. The intellectual landscape of 1953 was fertile ground for revolutionary discoveries. Prior investigations, notably those of Rosalind Franklin and Maurice Wilkins, had painstakingly elucidated the X-ray diffraction patterns of DNA, suggesting a helical structure. However, the precise configuration remained elusive. The work of Erwin Chargaff, detailing the characteristic ratios of nitrogenous bases in DNA, provided crucial clues, further emphasizing the importance of base pairing in the structure. These precedents formed the critical bedrock upon which Crick and Watson built their paradigm-shifting model. The challenge they sought to overcome was the precise arrangement of the constituents of DNA within this helix and its profound implications. Crucially, Crick and Watson integrated diverse strands of information. They were not just adept at formulating the structure of DNA, but also adept at synthesizing and applying existing knowledge of the structure of nitrogenous bases, the chemistry of sugar phosphate backbones, and the intricate geometry of the double helix. Their work meticulously detailed the arrangement of adenine with thymine and guanine with cytosine. Their model incorporated the principle of complementary base pairing, a cornerstone of DNA replication and functional expression. The precise mathematical relationships governing these interactions, particularly the specific hydrogen bonding patterns between the bases, were explicitly defined and are now considered as fundamental to our comprehension of genetic inheritance. Their model, articulated with a level of mathematical sophistication, clarified the mechanism governing the stability and replication of DNA, a cornerstone of the new era in biology. The direct applications immediately apparent were profound. The structure of DNA, as elucidated by Crick and Watson, offered a mechanistic model for heredity. The discovery provided the first clear explanation for how genetic information could be both stored and replicated. The very mechanism of gene expression, still a mystery then, began to take shape. This realization immediately spurred new lines of inquiry into the nature of genetic mutations and the mechanisms of evolution. The immediate and tangible impact lay in a new theoretical framework, one which empowered more accurate and targeted research in the field. The impact of molecular structure of nucleic acids extended far beyond its immediate implications. The work served as a foundation for countless subsequent developments. The understanding of DNA replication, initiated by this model, paved the way for the development of recombinant DNA technology. The discovery of the precise mechanisms for the transfer of genetic information allowed for the development of genetic engineering techniques. This transformed the landscape of biological and medical research, leading to the development of modern diagnostics and therapeutic interventions. Subsequent investigations rapidly expanded upon the framework provided by Crick and Watson. The mechanisms of transcription and translation, the processes by which genetic information is transformed into the proteins that execute cellular functions, were elucidated. Researchers built upon their findings to unravel the intricate complexities of the genetic code and to create methods for the manipulation of gene expression. The work fostered breakthroughs in fields ranging from human genetics to agricultural science. The conceptual framework provided by Crick and Watson has permeated a broad range of disciplines, including the burgeoning field of biotechnology. The contemporary reception of the work was initially a mix of excitement and debate. The elegance of their model and the clarity of their presentation were compelling, yet there were also questions concerning the role of other scientists in the DNA puzzle, particularly Rosalind Franklin. The ethical considerations of manipulating the genetic material were scarcely visible. Moreover, the initial interpretations and subsequent refinements of the model presented avenues for further research. This critical evaluation was a crucial element of the scientific process, driving further refinement of the model in the following decades. This rigorous scientific evaluation and refinement reflects the very essence of the scientific method. In conclusion, Molecular Structure of Nucleic Acids, published in 1953, stands as a landmark achievement in the history of science. Its insightful exploration of the double helix structure of DNA, meticulously presented, not only advanced our understanding of heredity, but also provided a foundation for a host of subsequent discoveries. The intellectual rigor, the precision, and the audacity of Crick and Watson's work had a remarkable impact. Their pioneering work continues to inspire and inform researchers today. The implications are undeniable. Echoing through scientific advancements across disciplines, this work represents a watershed moment in our journey to understand the very essence of life itself.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.